Hi, my name is Alex, and in this video, we're going to dive into the microscopic world of germs, or microbes. Microbes are all around us, and there are many different types. There are viruses, bacteria, fungi, and even parasites, and we come into contact with them every day. Though we often call microbes germs, many of them are not germs at all. We give the term germ to those microbes that can make us sick or cause disease. Today, we're talking to one of my friends and scientists, Amanda Mannino. She works with me at the Texas Biomedical Research Institute right here in San Antonio, Texas. Thank you for joining us today, Amanda. Can you tell us a little bit about what you do and what exactly are viruses?
Hi, y'all. My name is Amanda Mannino, and I'm a scientist and a graduate student. I'm studying to be a virologist. A virologist is just a person that studies viruses. Right now, I'm working in my lab. All of this really cool equipment that you see around me is what I need to study viruses every day. Viruses are super tiny microbes that you need a really powerful microscope just to see them. Basically, viruses are bubbles of proteins and fat that have either RNA or DNA inside of them. RNA and DNA are like little instruction books or computer codes that tell all the cells in our body how to live and grow. Microbes have RNA and DNA just like us. Viruses are important to everyday life. They infect almost everything that you see around you. You, me, plants, animals, and even some bacteria. We usually think of our immune systems as the things that help fight off germs. But as it turns out, some viruses are responsible for helping our immune systems to work. The viruses got into our bodies and left behind instructions that helped us fight off other diseases. So, not all viruses are bad.
Are all viruses alive? And how do they survive?
Some scientists are actually still arguing about this right now, but most would say viruses are not alive. We say this because a virus actually needs a host to replicate. Replicate just means making more copies of themselves. Also, they can't move on their own. Some viruses can float in the air or on little bitty particles in the air, but they can't move by themselves and need a host to carry them. A host is any living creature like a plant or an animal or a human that a microbe can infect. When viruses infect any of these living things, we say that that is the virus's host. For example, say that someone who is sick coughs without covering their mouth. Ew. They can spread that virus. The virus can then be carried through the air through tiny little droplets. If the virus lands on another person and enters their body, that new person becomes the new host for that virus.
So, are all viruses the same?
No. In fact, there are lots of different kinds of viruses, and they all have crazy names. Some viruses are really dangerous to humans, and that's why it's important for scientists to study them very carefully. We have different safety rules for different kinds of viruses. We call this biosafety levels, or BSL for short. Each virus is categorized into one of 4 biosafety levels, with 1 being the lowest and 4 being the highest. Each different level requires a special combination of personal protective equipment, also known as PPE. BSL-2 microbes are things like the flu virus and some kinds of parasites. These are things that can make you sick, but they're not usually deadly. Right now, I'm in a BSL-2 laboratory, and to be safe, I have to wear a lab coat and gloves. BSL-3 microbes are those that can be easily spread, especially through the air. Examples of BSL-3 microbes would be things like the SARS-CoV-2 virus, the thing that causes COVID-19, or a bacteria called MTB that causes tuberculosis. In some BSL-3 labs, scientists must wear a special jumpsuit and something called a powered air purifying respirator. We call it a PAPR, like this. BSL-4 microbes are highly dangerous to humans and are really deadly. A good example of a BSL-4 microbe would be the Ebola virus. Scientists must wear a fully contained suit in the laboratory with a special air supply.
So we've all heard a lot about the coronavirus or SARS-CoV-2. Can you tell us a little bit more about that?
Scientists discovered a brand new virus last year. It's called SARS-CoV-2, which is short for Severe Acute Respiratory Syndrome Coronavirus 2. I told you these viruses have crazy names. It's called this because of all the little spiky proteins that are on the surface of the virus, and it sort of looks like a crown, which is corona in Latin. COVID-19 is the disease caused by SARS-CoV-2. It's spread through the air in little tiny microdroplets caused by coughing, sneezing, laughing, or even singing. These droplets can be in the air or on surfaces like door handles. When a different person breathes in those drops or touches the doorknob, they can become infected and get sick. They can get a fever, cough, have trouble breathing, and in some cases, they may have to go to the hospital for over a week. Sometimes, COVID-19 is deadly.
Wow, that's fascinating. Seems like we need to do a really good job with preventing the spread of this disease. So how can we stop viruses?
The best way to stop the virus is to stop spreading it to new people. The easiest thing you can do to stop the spread is to wash your hands. When you wash your hands with soap and water, the soap acts as a degreasing agent that can break down those viral particles. Then, they completely dissolve the virus and it can be washed down the sink. If you don't have soap and water, hand sanitizer is almost just as good. Also, you can practice good social distancing, which just means staying home when you can or staying at least 6 feet apart from other people when you do have to go out. You can also cover your mouth and your nose with a mask when you're around other people. This helps to stop the drops in the air that contain the virus from getting on other people. Did you know that when you sneeze, those droplets can travel up to 26 feet? Gross.
What kind of work do you do with the coronavirus on a daily basis?
Right now, I spend most of my time working with coronavirus in a BSL-3 lab. We performed experiments with rhesus macaques and baboons, where the monkeys were infected with the new coronavirus, and then we studied what happened to them. We drew blood and also took swabs of their nose and their throat. Now we're doing tests on those samples to see what's happening in their immune system to fight off this virus. We're doing this to help create drugs and a vaccine that can help fight SARS-CoV-2.
Aside from COVID-19 research, why is infectious disease research so important to everyone?
Microbes cause millions of deaths around the world every year. Thankfully, advancements in nutrition, vaccines, food safety, and sanitation have led to a huge reduction in infectious diseases. However, they can still be a big problem for a lot of people. When a microbe enters the body, some people are able to fight it off with their immune system, while others can't. That's when people develop disease and get sick. It's important that we study infectious diseases because we live in an interconnected world. Since it's easy to travel all over the world now, a new disease can spread to any city on the planet in just a few hours. Sometimes all it takes is one sneeze to spread the microbes throughout the community. When diseases spread all over the world, we call them pandemics. The pan part of pandemic means all. Right now, since SARS-CoV-2 is all over the world, we are living in the COVID-19 pandemic. Science, education, and communication give us a chance to detect infectious diseases early and to take action immediately to slow them down before they can spread too far.
Thank you for sharing what you do, Amanda. Now, let's start the activity, shall we?
Hi guys! Good morning. My name is Alex Wadley, and I work at the Texas Biomedical Research Institute here in San Antonio. And I'm here with my partner in crime, Amanda Mannino, and we're going to go over the Micromania activity with you all today. So many of you should have received the take-home kits, which includes a Petri dish, Play-Doh, gloves, and some wax paper. So we've heard a lot about microbes. We've heard a lot about biosafety levels. And so we're going to ask you guys to model one of the microbes that you've received and put it in your Petri dish. Okay, so by this time, I hope you have your gloves on. And you know what? I'm gonna go ahead and put a face mask on. So I'm gonna take the loops here, place them around my ears. And I want to concentrate on this metal part at the top here. Now I'm going to pull it apart and pinch this little tiny metal strip around my nose. Okay, so now I'm fully covered from our microbe. Alrighty, so some of you should have received the influenza virus or the flu, and you'll find that on a sticker in the back of your Petri dish. The influenza virus is a biosafety level 2 virus. Some of you have received the COVID-19 virus, and that we consider a biosafety level 3. And some of you have received the Ebola virus, and that is what we call a biosafety level 4 virus. So I'm going to model the Ebola virus for you. Now, if you want to go ahead and take your Play-Doh, just gonna try to make it look like the virus that you received on your Petri dish. Now, not all viruses look the same. Some of them are ball shape, have little spikes on them. Ebola virus actually is kind of long and worm-like. Now, if you guys have any questions, please feel free to ask. Okay.
What is it like to work in a lab?
Let's see. Is Amanda here? Is she on the call?
I am on the call.
Oh, there she is.
Hi.
Hi.
So I work in the lab every day, and a lot of the times I start off my day going on the computer and reading about all of the other science that everyone else is doing so that I can see what the greater scientific community is doing. And I read their research, and then I say, well, they did this way. What if I did it this way? So for instance, with the coronavirus research we're doing, we saw that people were doing experiments with marmosets, which is another kind of monkey, and we were thinking, we think that baboons would be a better model for humans and coronavirus disease. So we did experiments with baboons. So we start off the day reading about other research, then we go into lab and start our experiments.
Hi, Amanda.
Hi.
Are you working on a microbe today?
I am. I'm working on COVID, or I'm working on SARS-CoV-2. So I'm starting out with a Play-Doh ball, and then I'm putting tiny, tiny, tiny little spikes on my virus with little bitty Play-Doh balls.
Oh, that's fantastic.
What is your favorite part about being a scientist?
My favorite part about being a scientist is I get to discover new things every day. So sometimes I discover when things are wrong. So I'll have an idea and I'll test it out and it'll be wrong. But sometimes I'll test out an idea and it'll be something new that no one has ever known before. And I'm the first person to know that. And then I get to share it with the world.
Do we have to wear a mask?
If you're going outside and you're going to be around people, people that aren't your immediate family, you should wear a mask right now, specifically because of the COVID-19 pandemic. Wearing a mask helps you from getting sick and helping others to not get sick as well. So yes, if you're going outside and you're not around your mom and dad and everything, you should wear a mask.
How do you find out if someone has if they are asthmatic?
If they have asthma and are asymptomatic.
Asthma.
Oh, if they have asthma. So SARS-CoV-2 is a virus that infects people, all kinds of people, but specifically people that have certain other diseases that makes them more susceptible or more vulnerable or more likely to get the virus. So some people with conditions like asthma or other lung diseases or diabetes, or if they're really old, they're more likely to get the virus. And they get the same test as everyone else. It's a really long swab and it goes way into your nose. Like it feels like it's going all the way into the back of your brain. And they swab in there and then they test it using a method called PCR, which is just a fancy method of detecting the RNA or DNA in that sample. And so everyone gets the same test basically to see if you're positive for the virus.
How long does it take to develop a vaccine?
That's it varies wildly. So sometimes a vaccine in development can take years. So for instance, when I'm not working on coronavirus, I do research with HIV and SIV, which is like the monkey version of HIV. So we've been working on a vaccine for HIV since the 1980s, and we still don't have one. So it's been 30 years, 40 years we've been working on a vaccine. However, some vaccines get developed really quickly. So every year there's a new flu vaccine because every year there's a slightly different kind of flu. The little proteins on the outside of the flu change sometimes. So they have to make a new flu vaccine every year. So that vaccine only takes 9 to 12 months to make. So big difference, anywhere from a year to 40 plus years.
About the germs sometimes?
I'm sorry, could you repeat the question?
Do you get scared about the germs sometimes?
So I work in a BSL-2 laboratory a lot, the one that you saw in the video. And those viruses aren't very harmful to humans, and they're really kind of hard
What kind of schooling do you have?
Let's see, can you guys hear me?
Yes, we can now. Thank you.
Oh, okay. Is Amanda still on the call? Or she fall off?
Hello.
Okay.
Yes.
Oh, there she is.
My internet's being weird, so I'll just be on my phone now. So yeah, so when I first got trained in the BSL-3, it was a little scary, but once you realize that there are measures in place to make you safe, then it's not scary anymore. You know that you're wearing the suit, you're wearing gloves, you're wearing all this crazy equipment to make you safe, and you get trained a lot. So after I was scared a little bit at first, it got better, and I'm not so scared anymore.
Speaking of training, what kind of schooling do you have?
So I went to UT Austin in Austin, to University of Texas, go Longhorns. And I went there for 4 years, and I got my bachelor's degree in clinical laboratory sciences. So that's 4 years. And then I did a training program where I learned how to do all the stuff in lab. That was another year. So 5 years. And now I'm in a graduate school to become a doctor in virology. And that's going to be another 5 or 6 years. So anywhere for me between 10 and 11, 12 years to become a virologist.
What did you want to do to be when you grow up?
So when I was little, I thought I wanted to become a medical doctor. So a doctor you go in and see when you're sick, or someone that does surgeries or things like that. So that's a doctor, and they have very specialized training as well. They see sick patients. But I wanted to work more in the lab. I wanted to go into lab, work in the hoods, work with viruses. And I didn't know that working in a lab was a separate thing for being a doctor. I thought that a doctor was what you did to work in a lab. So I went to school, and I was actually going to school when I was in college to be a doctor. And then one of my teachers told me, you don't have to go to medical school to be a scientist. You can go to graduate school, or you can have an applied trade like I did. I worked in a hospital lab for a couple of years before I went to graduate school, and I didn't even know that was a thing. So I wanted to be a doctor, but I really wanted to be a scientist. And I'm glad my teacher talked to me and told me those are two different things.
What sparked your interest in biomedicine?
This is going to sound silly, but I read this book when I was little. And my mom got it for me. She's a nurse, so she always bought me science-y or doctor-y kind of things. And it was this book called The Hot Zone, which is about basically Ebola virus and how it spreads all across the world. And I thought, wow, that's so cool. These doctors go out and do all these tests during this really dangerous time, and they can get answers, and they can help fix the problem. So that's why I wanted to become a scientist. And that's great, because now I'm doing that. I'm going out, doing all these tests on coronavirus, this thing that's in the world that's infecting people. And hopefully our work can help fix that.
Do we have any more questions?
Do you
For
love
Amanda?
working with the virus?
I'm sorry, say one more time.
Do you love working with the virus?
I do. I do it every day. I like going into lab. I like knowing that I'm one of the few people that are trained and have, the ability to go in and do this work. That not everybody can do that. That's cool. Like doing something that no one else can do. That's so awesome. So yeah, I like going in and knowing like, hey, what you're doing, not a lot of people can do that. That's so cool. So yeah, I like it. I like going in, working with the virus, putting my gloves on, doing all my pipetting, really learning about the virus. I think it's really awesome.
Thank you for everything that you've done for us.
Thank you. Thank y'all. Thanks to Girls Inc. for setting this whole thing up. I really appreciate it.
Are there any last questions for us? No? Amanda, I have one question for you.
Yeah.
What would you say, how would you inspire other girls to achieve a STEM education? What would you say to a young lady right now to want to attain a better future in STEM?
I would say that girls are just as smart, if not smarter sometimes, than boys. And girls see things differently than boys sometimes. So we need boys and girls in the lab to really think about how to solve certain problems. If we didn't have girls in lab, we wouldn't have certain discoveries. So for instance, Rosalind Franklin, who is the girl scientist that's on my shirt right now, she helped discover the way DNA is shaped. And if she didn't discover the way DNA is shaped, then I couldn't do my work in lab. And you couldn't get your PCR test for COVID-19 because it tests RNA and DNA. So without girls in lab, nothing gets done.
That's our
Well,
last question for now.
guys
so
and girls,
thank
thank
you
you so much
for your time.
for joining today. So excited to teach you about COVID-19. Just remember, stay bold. If you guys went to Texas Biomed website at texbod.org, us up on Facebook. Alrighty. And
Thank you so much for your time today.